Hello everyone, thank you very much for attending my talk. My name is Maryam Nazarieh and I will talk about identification of somatic stem cell specific functional units. Unlike bacteria that can be deactivated by the antibiotics, viruses evolve in the host body, cause a constant effort to eliminate them. And the problem becomes worse if they target somatic stem cells. Consequent infection process can transfer them to cancer, where the cancer stem cells are resistant to drug therapy. To address this issue, it is helpful to identify the stem cell either in the case of immunization or for the treatment based on identification of organoids which are derived from stem cell by image processing, and then identify the somatic stem cell specific functional units which played a significant role in transferring them to cancer stem cells. To identify functional units for the purpose of immunization based on pattern recognition, having expression data, we suggest to transfer biological modules from well-annotated biological networks by solving click-based graph alignments. And if we have medical image data, we suggest to work on the algorithms which maximize the score of matching between the trained dataset and test dataset, and consider the computational methods which return connectors in the set of results if we have transcriptional networks. To transfer biological modules from well-annotated biological networks, we suggest to follow click property, where two nodes of two graphs are taken into one click if they have less dissimilarity than a specified threshold. To match the training segments to the test dataset, I developed an algorithm which consists of 3 steps with quadratic time complexity for the exact matching, and to identify the key player genes in transcriptional networks which transfer the stem cell into cancer stem cell. We suggest to consider the minimum connected dominating set which returns dominators and connectors as the results, with focusing mainly on the connectors for embryonic stem cell, and path the fate for the somatic stem cell, such as blood stem cell. Blood cells have a specific expression pattern. As I have displayed the expression pattern of identified transcription factors in 6 stages, starting from embryonic stem cell until macrophages that follow the global expression pattern. As you can see in this slide, the expression pattern in the first 5 stages converges, but in the last one, which is macrophages, diverges. And at this point, it is postulated that the virus evolutionary path complies with the evolutionary path of the blood cells, and by circulating blood, the connectors of the virus evolutionary path play a significant role in spreading the disease to the organs for the maximum efficiency to survive. Since virus infection can infect brain, cause memory loss, disability to do personal activities, and also visual illusion, it indicates that high distance between reality and illusion can be a sign of brain infection. So to immunize brain organs, first it is necessary to check the healthy status of a spinal cord by relying on the deep learning models over the medical imaging datasets. And then to identify the biomarkers related to brain immunization, we suggest to solve a dual optimization problem such as Parkinson's disease progression to identify the biomarkers related to brain immunization as a primal problem. If a person suffers from chronic diseases as well as virus infection, we suggest to follow the personalized medicine pipeline for combinatorial diseases, which consists of 6 stages, starting from image processing until identification of biomarker genes such as biomarkers in top control methods and identification of biomarker underlying and related to DNA mutations. In order to check the effect of virus in cancer metastasis, which is also one of the combinatorial diseases, I developed a deep learning model consists of 3 layers such as large bowel, a small bowel, and a stomach, and connected these layers through Donkey theorem, and it worked and changed the results remarkably, which can indicate the maximum efficiency. I applied the deep learning model and the algorithm to Kaggle competition projects, and I obtained remarkable results. In conclusion, I would like to say that identification of set of functional units either for prevention or the treatment can be addressed from multiple aspects, and level of confidence for the suggested set of findings will increase if it is the outcome of multiple approaches such as network and expression analysis or image segmentation analysis. Thank you very much for your attention.